رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني. إن شاء الله تعالى I'll see you soon. Inshallah ta'ala, students will be doing the weeks 9, class 2, microbiology, on analyzing environmental conditions of the bacteria, and doing some plates for differential plate streaking. So seeing different types of traits of bacteria based on selective medium or differential components of it. So inshallah, I'll see you soon. Barakallahu. Stay tuned, inshallah, ta'ala, for second last month or second last class. Or powerful power.
If we have unlabeled tubes, right? We have unlabeled tubes to blank or something. Yeah. Oh, I guess it's funny about it. It's sort of about I think last week they were such a results. Okay. It's like yes.
Yeah.
No,
I think we're just and then I would have
I want to give it a vote.
like fighting.
So the specific that on labels, this is the one that has the red list. We'll give this to the other ones already. That's we can figure out. And maybe review.
Sounds good.
Oh, yeah, literally before I.
On the other end?
Yeah. So I like to talk about like, yeah.
Oh, she's left. Yeah. I'll take it.
Yeah, yeah, I was like, it's maybe. I was like, I'm not literally. How about you? I maybe. I love the bunch of there that like, yeah, so now I have focus on like, you know. Find somewhere. I live like. Six, but I can't be. Oh. Last time you played like 5 cents. Because I also. On the kind of it.
That's what I'm just thinking. I think this is really
Hello, everyone. So we're here now in our second last lab, the second last class.
That speaks a little bit.
No,
It's an actual.
it is a lab. It is a lab. You always, so, I mean, you are looking at your result. Let's see. Yes, so I'll tell you guys what you have in due time. Um, Let me just actually tell you what you have. So you guys have three sets of plates for exercise 1, 3, and 5. One is your MSA plate, Mannitol Salt Agar plate. That's exercise 1. It's a more darkish red tinted plate. You have for exercise 3, the McConkey Agar plate. That's a bit more pink and selective accordingly. The Jazakumullahu test tubes are on the far left for those on this side, far right on that side. It's labeled. There's like sets of 18 tubes, 3 per. If anyone, by the way, is feeling sick or ill or needs a band-aid or anything or needs to leave to use the washroom, just let me know. I just really appreciate being known because otherwise I don't want people leaving in and out without me being aware. So just bear that in mind. But of course, like I don't hesitate to let me know. The last plate is your blue one. That's the DNase plate. So before you begin, I recommend, or inshallah, what you could do is actually cut out those sections. Not literally, but like using oh, I hope. Okay, so the idea is that you technically have 3 sections for all plates. If you want to do like a fourth, that's okay. Just let me know. Because you have more than 3 bacteria or more than 2 bacteria shown from the specific slides. Um, And just make sure that you label, right? Exercise or practical, the group, practically. Let's say your group 1, PR1. Exercise, it could be 1, 3, 1. The different specific bacteria or conditions could be, for example, E. coli. This could be Staphylococcus aureus or epidermidis, and then this could be your control, which is normally going to be actually negative because you wouldn't have bacteria present, right? So that's something you want to keep in mind. And similarly for the gelatinase test, you have 2 bacteria. The solid agar, yes, the nutrient agar slants that you'll just place in with an inoculating needle into halfway into those respective tubes. You have 2 bacteria. One will serve as a control, so you don't have to touch it, but you just label accordingly your plates and tubes to the best of your ability with the details I've described earlier. Any questions? That was a more fast-paced intro. But generally speaking, as you can see from the top, there are different components of media and different techniques that get involved for microbiology. And as we complete this course together, inshallah, what I want you guys to appreciate is that there are different selective and differential components. What's the selective component? Does anyone know? Anyone know? Okay, I'll just mention something. So I also am tight with time. So okay, so you can see here, to make it easier, the biochemical tests, right? They refer to specific modes of being able to identify or characterize traits biochemically of your bacterium of interest. For example, the catalase test, we're able to observe bubbling, and that indicates the level, relative level of catalase and enzyme that is used for productive or antioxidation, because essentially it helps combat redox or reactive oxygen species. So that's one example. The beta-lactamase or cephanase test, if you recall, that was able to elicit, or what we were able to appreciate are the presence or absence of beta-lactam. The beta-lactamase enzyme, which breaks down beta-lactam in different antibiotics. So think about these different properties, techniques, and actually this will help us move forward to Samah, if it's okay, can you close the door? I really appreciate it. That way, if it gets more warmer here, you guys don't have to feel too. But essentially, we'll also look, inshallah, at week 8 results. I place them right beside those tubes that are more see-through, right? And so as a group, as a lab, we're actually going to look through the different tubes for week 8 here. These are the Jazakumullahu test tubes. And these are your tubes, or your plates for viable counts last week. I think the group has been mixed with more than 4. You got this? You got this? Perfect. So what I'll just recommend though is when we're at the stage of week 8 results, we'll do them as a lab, or we'll look at them as a lab and try to compare our results accordingly. If we have time. I just feel like there are procedures that may take a bit of time today, so that's why I want to go through them efficiently. But let's see. Mannitol, McConkey, the gelatinase tests for exercise 4, once again, are those tubes. And you're just puckering or like poking halfway deep into your tube. So it's similar to the motility test, but you're just going halfway in. And then in contrast, for exercises 1 and 3, you're making smears, or like streaks essentially. So bear that in mind. You're not just doing a spot inoculation because you want to see the changes in your results post-incubation next week. And this is, yeah, so then biofilms, This will be done by the lab tech hopefully next week, so you don't have to worry about this. Likewise for the Snyder test and the blood agar, we'll talk about them as a group. But in the meantime, let's go forward with, let me see. There is a component that I need to talk about. I'm just trying to see if we have the time to, and that is for the different ingredients. So we have selective and differential components of media. Selective will help you discern if bacteria grow or not in your media, right? So think about ampicillin resistance, right? From the transformation assay we had in like week 7 or 6, and you were able to see some bacteria grow, indicating that they are ampicillin resistant. That indeed is the selective marker, right? If you recall. And similarly, it serves as the selective component of your agar. In this regard, you have other finite or specific ingredients that also help discern whether your bacteria can be selected for or differentiated. And when it comes to high pHs or low pHs or high environmental conditions, so like essentially going to the extremes as what we've done from the past, you can appreciate that those are generally selective or depending on the assay, and I'll talk about it now. In the case of NaCl, this is a high concentration of salt, table salt. And indeed, the idea is that bacterium that are osmotolerant should be able to grow here. So generally speaking, based on the two that are shown, E. coli versus Staphylococcus epidermidis, which you should recall have differences in gram staining, right? That would in of itself serve as a unique step for differentiation. So differentiating, bacteria based on specific traits. To make it easier, let's actually just go ahead. Let me see. Yes. So when it comes to like phenol red, dyes, colors, methylene, methyl green, bromocresol green, as we did with the capsule stain, with the endospore stain, you have like essentially those dyes to be able to differentiate, bacteria like Staphylococcus epidermidis versus Staphylococcus aureus. So these are things that you want to compare. And generally speaking, as you see changes in color, those exhibit differential components. And that could be exhibited by lactose, which helps ferment, right, your bacteria, and thereby change the color of your media from neutral red to a hint of yellow, golden orange, or with the crystal violet, remain in purple. And so these are the different colors, as you can see here, are differential. They exhibit a spectrum of how your bacteria are able to grow. In contrast, if you don't have bacterial growth, which can be seen like for the DNase hydrolysis test, that exhibits a selective trait. Because if they're able to grow, like if, let's say, for example, you have, actually, let me just clarify this. The clearing, yes. So the clearing essentially would help you exhibit essentially that the bacterium of interest, even though they can grow, they would essentially have the DNase essentially clear out regions of your bacteria or regions around your bacteria, making a halo as you saw from the capsule stain. So in this regard, this is selective. But you just want to bear in mind that, technically, and this is something that you want to bear in mind, the ingredients are important. So, like, if let's say this plate was at a pH of 4, right, that would make it a selective component of your plate. If all bacteria are able to grow, then generally speaking, how you differentiate them is by a differential component. So that's like in a nutshell. Maybe just to go straight ahead, are there any questions? It takes a bit of time, and I actually don't want to delay, rather. So go with what you see in the manual, essentially to make it simpler to understand. as that way you're able to go back and revise the concepts that you'll learn for today. So once again, make 3 sections for each of the plates. Your group members can essentially split up accordingly. Make sure your plates are labeled. And inshallah, the gas is on, so you guys can, your 4th group member, if you have one, they could start the Bunsen burner. And just once again, remember aseptic technique as you complete exercises 1, 3, and 5. And so I want you to do this in the next 20-ish minutes, 20, maybe 25 minutes. I'll try to let you guys know. For timing, try to be done by 10 is my advice. Any questions, by the way? No questions? No questions? I didn't mention this as well, but for those that are wondering for your FLRs, time, do I have the time to? Okay, you can continue getting started. I'll talk about like some other updates to save you guys the time. First off, when it comes to your post-lab, folks, please make sure you have detailed figure captions. So some of you are asking or were wondering perhaps, what's the figure caption looking like? You want to have a title for each experiment. So for example, your plaque assay determining the number or the original cell density or bacteriophage density using the plaque assay in specific details, E. coli with the lambda phage. So you put specific details, specific terms that are important, and that's what I recommend you doing. And then thereafter, highlighting the methods, highlighting the conditions that you're working with. All of your tubes for exercises 1, 3, and 5, by the way, should be labeled, right? So only use the tubes that are labeled for a specific exercise. If you're looking at exercise 1, it's E. coli and Staphylococcus epidermidis. So you should have tubes that are liquid, and you'll be using the sterile cotton swabs. So you don't have to use an inoculating loop for this one. Focus on, collecting. And don't forget resuspensions. Resuspensions, folks, very important. Yeah.
Size 3 is not spot inoculation. So
right.
But the other one is
all of them are not a single spot inoculation. It's just the strategy that comes into account. Exercise 1 and 3, you're streaking across your section. Whereas for exercise 5, the blue DNase hydrolysis test, you're making a single streak, like a single line for each condition. That way you should be able to see if it's a positive result, a halo surrounding your back. And any questions? If there is, so in case there is like a delay, actually let me go to today's. Yes, I did. Sorry if I, because I do get a bit tense. Most of the time, like I get different things. If I like action items, but do know I have not like I did. Any questions for today? No problem. And when you're done those exercises, let me know. You don't have to go forward to the other ones as of yet. So just try to do exercises 1, 3, and 5, and let me know when you're ready for week 8 material or the gelatin test. You could go to that one if you're ready. Yes. All of them. All tests, all plates need 3 sections because you're gonna have an uninoculated control section. I know, but you guys want to be, so as scientists, you want to make sure you can bear in mind controls. Bear in mind that, like, for example, if your bacteria were contaminated, you may actually see inconclusive results. course, but it's low. It's low in what do you need it for? Don't worry, I can help you. And the other control, so your other last section will be your control, and the other two bacterial you'll have in the other thirds. Just before?
Do you have any more sterile console?
I do, but how many do you need? Because technically,
We only have one job. Yeah, well, we only have one job. Are you?
He did it too.
any questions, folks? No questions? Use for all plates. Use your liquid drop. You should have it for each condition. Let me know if you're missing a bacteria. Someone put some burners on a bit fine. Yeah.
So for the gelatin, we use the solid methods, right?
Yes. I like how you guys are paying attention and focusing. I do notice some of you are maybe on your phone, so just make sure that you're not, be mindful actually. That you want them to receive your laptop. If you maybe use your phone to take photos or answer your questions.
My mom's Yeah.
Yeah, that's.
They're all. Yeah. Yeah.
Okay. And then expose How are you guys doing? Thank you. So, you've done all three plates. I just want to get an update. Done all three plates. Make sure that you tape them, label the tape, so that way you can place them on the top left of that incubator. Both top left incubator at the very top. Bear that in mind. We're not using the bottom one. And similarly for the Jazakumullahu test for incubations, we're going to put at 37 degrees rather than 25. We're doing it at 37 degrees, and we'll place it as a move into a single tray. You think bear that in mind? No, this is top. Top incubator at the top left, and then focus on the Jazakumullahu if you're not done. Thereafter, we'll talk about remaining week 9 and week 8 material. To your plates now. Just to confirm, all groups are done. Exercises 1, 3, and 5. All groups. I want to make sure you guys are done. You guys are doing 5. Okay. So you're doing 4. You done? So groups. Please make sure that you've labeled the tape surrounding your plates. The 3 plates. You would have inverted them and placed them inside there. In the top left incubator. When you're done that, there's one more group. Please make sure, folks, that you bring it once again, and that each the incubator will put in the Jazakumullahu increasing box. See that's on the yeah. Only on the top left. So okay, let's do some all.
like that.
Group 4, if I can have your plates. Your 3 plates, if they're not in the incubator, there's one group who hasn't put it in. So if you haven't, please do so now. Let me now briefly go forward. You guys are ahead, just like a minute or so behind, but that's okay for timing. Jalanese folks, let's pay attention. So I didn't talk about this, but for understanding the concepts, this is another type of enzyme that ideally would be selected for based on your agar and its viscosity in a week or technically a day's time. The idea is that if you have gelatinase in your bacteria, it would
In your vertebrae.
in your vertebrae.
Yeah, I think that's what I was writing.
Not necessarily. But so the in your vertebrae is more actually more. It does, but that's a good point. But at the same time, you don't want to. I don't want us to exaggerate. Like I say that because okay, the solid. So the vertebrae for us is actually relatively more solid. And I mean there are components where, especially if you have high densities of gelatinase, the idea is that to answer this question, gelatinase would essentially break down gelatin found in the gel, or essentially in your tubes. And so as you poke inside each of those tubes, if after a day, at 37 degrees, you find the tubes to be viscous, then that indicates a positive result, i.e. that you have gelatinase in your bacteria. And that can connect actually to the more functional or physiological effects because it can, but folks, let's pay attention. Pay attention, folks. I just want us to bear in mind that physiologically there are many stringent operations in your bodies that makes it quite difficult for even these types of bacteria to break down your vertebrae. Or there are maybe pigments like very like where there's soft ligaments, for example. That's where essentially if you have edges, I don't want to go on a tangent, but essentially there is the potential of minor breakdown. Yeah.
Very bad.
I mean, yeah,
Like
hypothetical,
bad.
but hopefully not. Hopefully not at all, in fact. So let's just briefly go over this once again. If you have a positive result, then after a week, when you come back for week 10 and see your results, you'll be able to like wiggle around your tube. Whereas if it was, let's say, tube B, then you do like a Dairy Queen flip, and you shouldn't see the agar or the gel flop out. Hopefully, that's essentially your negative result, indicating that you don't have gelatinase, that enzyme that would break down the gelatin in your agar. So bear that in mind. And similarly for the other results, just to highlight this once again, this is a selective component, and bear in mind the methods because essentially you have specific ingredients that are not mentioned here, but are noteworthy for understanding the different components and whether or not they're selective or differential. Once again, differential components essentially showcase bacteria growing in different colors or in different ways that are exhibited by color or different colorations, whereas selective components usually show a like bactericidal effect if they grow or they don't grow. And so that's something that you want to bear in mind. How do different ingredients play a role in differentiating or selecting the growth of bacteria at all? And that also hopefully will give you an understanding of all 4 exercises that we've talked about thus far. Any questions once again, or no? Questions? 0 questions? Okay, so biofilms, once again, we'll get these results next week, hopefully, inshallah. The idea is that you start off with a plate. It's actually very simple. in that you have like, think about all your liquid broths, right? Imagine you kept resuspending them, but they were growing and essentially propagating in that single tube at 37 degrees. Thereafter, what you would do for Staphylococcus epidermidis is essentially rinse off, not rinse, decant any residual, like supernatant. And because the idea is that the bacteria is going to actually surround the surface of your tube. And that's where our biofilm comes in, because if you're interested, let's say in medicine or pathobiology, the idea is that biofilms make up not just bacteria, but they're secreted products and extracellular extracellular layer essentially, or extracellular matrix, known as ECM for short. And that includes like sugars from like the glycocalyx that's also attracting certain nutrients for bacterial growth. And so you can have like amelia, meaning like many different types of bacterial species, all being collected in this community of bacteria found in a population of bacteria here in a biofilm. And this has, impacts not just like in the slants, but in more in other uses as well. But for day 2, which is what you'll see next week, inshallah, is essentially dropping it down into the Wescadine, rinsing it with water, PBS, and then staining with crystal violet. This will be a differential mechanism, in fact, like what you've done for the gram staining. You saw purple for positive, pink for negative gram stained bacteria. And so the idea is that if they're gram positive, Staphylococcus epidermidis, then you should see a generally purple coloration, kind of like what you would have seen from your slides at surface eye level. And then if you have lower concentrations of your bacteria, then thereby you would have a lower success of seeing bacteria secrete around that biofilm, and thereby reduce their propagation and indeed reduce the coloration of your tube. So just bear that in mind. Tube D has a greater propensity of bacteria in your biofilm, and it decreases all the way to tube A. Yes.
For the virtual.
I'll talk about that. Not right now, so that way we could go ahead with other things. But I do want to reserve a time for you either this week or next week to go more into the lab exam, or actually lab review. I want you guys to be fully prepared, inshallah. So let's focus on the slides for now. Any final questions, folks? Okay, so Snyder test is actually very important for your lab exam. So I mean, at the same time, Dr. Brunt is like, I'm not making your exams, right? So just bear that in mind. Dr. Brunt may make edits and changes. So no, I haven't for this term. No, can I? I may. But nonetheless, Snyder test is essentially okay, so I have one more. So okay, the idea is for your Snyder test, imagine doing a swab in real life without any bacteria in your tubes, but on your hands, in your teeth, with your saliva. This is actually what you're doing with the Snyder test, in that you're actually taking regions of your molar, like pretty much molar regions or maxillary regions of your teeth, and thereafter, essentially, you could also take like a cheek swab. But the idea is that if there are lactobacilli or like bacteria that ferment lactose, then they should actually lead you to see a change, inshallah, in your coloration of your tube. So isn't that amazing? Like imagine you do a cotton swab in your mouth, maybe even do a rinse, and thereafter place that swab, the same swab, with aseptic technique into your tubes. And then you should actually see a change in color over time. Maybe it'll take a day, or a few moments, maybe multiple hours, but nonetheless, you should see a differential change. I do want to highlight though, there are three main ingredients or core ingredients that you want to bear in mind. Dextrose, glucose, these are metabolites, sugars. Many of you are probably aware of this. And so they help for the metabolism of lactobacilli. And so what you want to bear in mind is that for them to grow, then this would be a selective component. In the case because of you seeing qualitatively, right? Because you want to see a change in color. So if they grow, then that change in color. So actually think about it like this. The bacteria, if they're present and they're lactobacilli from the different species that exist, you would see them ferment glucose or dextrose. And thereafter, fermentation, for those of you taken biochem, would essentially lead to a drop in pH. Folks, let's pay attention. I can tell who isn't, so just bear that in mind. pH levels will likely change, right? And with the changes in color, then you would be able to depict the concentrations relatively, or the abundance of those lactobacilli in your teeth. And that's going to be a bit similar to what we're talking about thereafter for week 8, because you want to see changes in turbidity. And that will give you a marker qualitatively as to what your readings should appear like. So this is a good example, right? So comparing, let's say, tubes 1 and 3, rather than tubes 2 and 4. Tubes 1 to 3, which one would have more lactobacilli? Anyone know? 3, yes? Why? Yes, and can you elaborate more? Because 5 is the most high, but I was just saying between 1 and 3.
So it's forcing.
It's not just a breakdown. Something to point out is that there's more bacteria. So if there's more bacteria, then there'll be more breakdown of the even a finite amount of sucrose or dextrose or glucose. So it's not about that be like that there's more sugar per se, but there is more unless like the experiment was inverted and you actually had bacteria in there and you were just actually just seeing the amount of sugar content in your teeth. That's a bit different. But the idea is that yes, pretty much they are more turbid based on the fact that you have more, sorry, they're more yellowish green, more towards yellow because, sorry, I didn't want to take your shine, like take away your speech. So, but the idea is like, it would be more yellow. Garvin, you want to say something? Yes.
Considering that it just turn over glucose faster, like efficient output glucose.
But so that's a good question, but at the same time, you want to bear in mind like reactants versus reactants and limited substrates. So like in this case, your bacteria is one primary reactant, and your substrates like sucrose or dextrose serve as limiting reagents. Or actually, let me say it in another way. All of them should be controlled to have the same amount of glucose, for example, right? Or sugar. What differs is the amount of bacteria. So you want to bear in mind that when you're taking these swabs, for example, the bacteria themselves, if they're at higher like proportional levels, if they increase, if you have a lot in your saliva or in your teeth, then they would correspondingly lead to a greater hydrolysis of dextrose or glucose, thereby greater fermentation or metabolism, and thereby a greater decrease in pH, i.e. more acidic, and showing a greater change in coloration from dark forest green to more pale yellow, looking like lemon acid yellow. So that may also help you understand the concept in terms of change in color. But does that answer any outstanding questions? I may have to skip ahead. Let's talk about blood agar. This is, and actually, let me know if you guys want me to slow down. I just want to bear in mind that I want you guys to understand the concepts as well. But if you have questions, don't be shy. I'll be happy to help out. Blood agar is also very important, and is differential because you're seeing changes in color, right? You're going from yellow to beta hemolysis, hemolysis, because they're spelled differently. You have HAEMO and HEMO, but the hemo refers to hemoglobin. So think about like RBCs, red blood cells, have hemoglobin, and so what these different bacteria like Streptococcus pyogenes would be able to essentially incur the destruction or lysis of these RBCs. And thereby you'd see a change in color that for beta light hemolysis, you would see essentially yellow, a clear yellow tint or clear yellow streak across your bacterial colony or bacterial spread or streaking pattern. So that would be more yellow on the left-hand side for the figures that you see there on either panel. Whereas alpha is more, it's less in degree. And something that I want you to keep in mind for your multiple choices is, or even just your understanding of the lab is essentially rather the modes. The modes of hemolysis are very important. So you have like streptomycins, like you have student like hemolysins, for example, enzymes that break down RBCs in specific ways or specific modes, and you want to bear in mind how that occurs for each of the different categories. For beta, which is more yellow, alpha, which is less yellow, and then gamma, which is generally speaking indicative of no hemolysis. You would want to consider though, like how you'd be able to discriminate those other bacteria if they're in the gamma hemolysis category. Does anyone know? Like, what would you do if you just wanted to know if they have other separated traits? Like, what would you try to differentiate? So, like, let's say you had multiple bacteria here who have hemolysis, and they all appeared like negative. What would you want to do? Like, what could you do to try to differentiate? That could work, but you wouldn't really see too many signs to differentiate them for a gamma hemolysis, or even just like separately. I want you to think of like any exercise you've done from the past.
We kind of like separate individual columns.
Separate different cultures of bacteria. A hint would be use another plate. So use a clear plate like the ones that we've done from the past. What I mean by this is because it's harder to see here with the media itself, you using rather than sheep's blood, right? Because that's where you see the red coloration. You would be able to see with other, like pretty much the plates that we have here today, the differences in their colors more directly because they're relatively more transparent. And so the idea is that bacterial growth without with the reduced contrast, you should be able to see more if there's contamination, different types of colonies accordingly. But nonetheless, just to highlight this, bacteria can still grow in gamma. It's just you wouldn't see, it would be more transparent. It's just, and that's pretty much why you want to consider the spectrum of colors as depicting a differential component of this assay. That's where I want to pause. Sorry, any questions, folks? Let's think about that because I don't know if Dr. Brun, I don't want you guys to take my words to heart because, or at least for now, because some of them, like Dr. Brun may change the exam. Yeah, so don't think that everything is the same, right? Like I took this course during COVID, so yeah. It's likely a mix. I'll talk about, unless Dr. Brun changes things, let's reserve the lab exam to the end of today, and we should have more time next week. Any other questions? You guys are going to need to do two things. Let me just see if there's anything else. Oh, Jelani's test, all groups are done? Exercise 4? I see 1, 1, 2, 1, and that group is not done. So what we can do, when you're ready, place them on this side, and then I'll place them in the. Now, in that time, as everyone comes in, you guys want to actually look at your tubes. Recall that they're all in different conditions for last week. Environmental conditions, pH, salinity, and now what I want you to do before you begin is recall some of them may be controls, but some of them, as you can pellets, right? What do you do? The idea is like you want to bear in mind some of yours may also have the lids covered well, but you want to resuspend well. Think about all these concepts in light of what you've done from the past, and you should be able to compare the differences in colors here. You could grab your tubes. This is what I want you to do. Grab your tubes and observe them after you resuspended them well. Make a scale from 0 to 5 as to how turbid you find your colony. So 0 is like not turbid, perhaps actually a good reference would be a control if it doesn't appear to have any bacterial growth. And 5 or 3, potentially compare turbidity readings. And you want to do that for each of your group's experiments. You just write something. Not turbid and high. Or you can say high and low. And so you want to bear that in mind. I want you guys to do that now, and then we'll talk about other things. We'll do things stepwise. And I want you to first just take out your tubes. And if you can as well, actually, this would make it easier. Take your tubes out for only osmolarity to determine the spec 20 absorbance readings. All other ones, you just have to look at qualitatively. So your groups are going to do the following. Grab your tubes, place them into your racks, resuspend them, observe for all groups, pH, temp, salinity, the differences in readings, qualitatively, just like from 0 to 5, and turbidity, and then only for osmolarity, i.e. the one that had changes in NaCl concentrations, that's the one that you want to put into the spec 20 to see more quantitatively what the results look like. Bear in mind that your spec 20 needs to be an absorbance. So bear that in mind because otherwise and by the way, blank in advance, and you could also use the Bunsen burner to flame the lids of your bacterial tubes. Any questions? No questions? You guys are doing good, by the way, so keep it up, keep it up. You can begin when you're ready. I'll let you guys know which colorful ones probably love for all of them you want to record your values for qualitatively. This is the turbidity. Yeah. For all of them, you want to do qualitative observation of humidity and then observation. And then just bear in mind once again, osmolarity, you want to make sure you find. And then record your observation. You want to rinse out your tube as you have the side tube. And you only have 4 tubes, so you're only going to do it twice. For the chemical analysis, I would recommend grabbing PayPal. Yeah, we're going to talk about it. But I want you to focus on you and then look for the different
Okay, what is that? This is three days, but one of the last.
I didn't know you were like.
Zero. But it's still 1 to 5. It's still 1 to 0 to 3.
Is that understood what you need to do? Like, you're recording the qualitatively turbidity readings from 0 to 5 for all of the experiments. I'm more precise. I want to have more wider range. You can do a 1 to 4 as well. The idea is that you want to get the spectrum, and we're going to come back. Actually, let me ask, how many of you have done it from 0 to 5? Okay. Any of you did 0 to 5? Because that way we can any other groups? Oh, you haven't started yet? Okay, so try to get ready, folks. We'll use 0 to 3 to make it consistent. But I want you to try to resuspend and take those values accordingly. And let me know if you guys need help in the next 5 minutes. Yes. You can do it before, after the illness. But my advice is make sure that you have the observation reading for spectrum. Sorry, qualitative. Yes. Yes, so folks, for osmolarity, you're doing both quantitative and qualitative. Please do the qualitative observations to scale from 0 to 3. I don't want you to throw out your osmolarity tubes because thereafter you wouldn't need them for your absorbance values. So just try to record the values. Try to do it in the next 5 minutes, and I would really appreciate that. Thank you. Yes. Sorry. 0 to 3. I said 0 to 5 because I prefer things to be more precise and to give a big spectrum, so that way different variables can be accounted for. But if everyone goes by 0 to 3, then that also makes it easier. Correct. No problem. No problem. Yes, garbage. Oh, you don't actually need the pipette. I mean, I don't like that. I actually. Oh, it's lost. So what we have actually to do is when you're done for osmolarity, because you have two cuvettes, first make sure you blank respectfully. Let me know if that's a problem. After you're done that, use the same blank to be able to record your values from the control, and then your other cuvettes for the other bacterial condition. Thereafter, you're going to rinse them with 70% ethanol, let them air dry, and then place the other two bacteria in the other two cuvettes, and that should be done for week 8 material. The last part for week 8 though is the TDC value, which we'll talk about. But I'm not, trust me, you guys are ahead in time. So even though I'm talking a bit quickly, just try to bear in mind time is what I would say. Don't rush too quickly, but take your time. You might as well unexpected ones here as well. For those that need it.
Yeah. Yeah.
It's all all the so it's all fair components, but they're all different functions. 5, 8. Yeah, that's your this one is only here. Yeah, it's just that the response creation. Is there control or not? No, the control.
Oh, I see.
I did 0.001 because it would go past.
Yeah.
One was one, that one.
Did you guys want to dry out? I don't want, okay. You can use this one for your control. and your other bacteria, and then that would be the final time for their yeah.
Because you already like made it from like the measurements. So you can use that to control.
You could still keep it out. But really, or you have already conditions, and then the idea is that you
You should alter.
Like 3 or 4 times. No, I really have to. It should be one. That would be. That should be. Just as an update, how are you guys doing? Have you all been able to record your values for absorbances? I heard from group 4, are you guys unable to? Okay, how about qualitatively? Please let me know if your group has not been able to qualitatively observe the different turbidity readings. And turbidity readings qualitatively, looking at the different colors or turbidity of cloudiness of your tubes. We need to do this in the next minute to finalize it, and then the spec 20 readings for osmolarity we could do thereafter. Yeah, for spec 20. Sorry, not spec 20. Okay, how about, okay, yeah, try to be done the qualitative observations in the next minute, because I want us to do the spec 20s thereafter. By 11, it should be done. You can, but I just because these two bets are sensitive, I don't want them breaking, and we may have a finite amount of them. That's okay. That's okay. Or you can try to do it like. Yeah. One other thing though, if you guys are done, tell me what you observed, and then PDT. Actually, don't worry, you're gonna like go ahead. But
I auditioned for UTSC and I think sports high school. Those brothers.
Yeah.
I think they were like, but they saw
But then just pass it up, right? I don't go before you guys. Yeah.
Folks, let's just get a recap. Has everyone done semi-qualitative observations? Okay, maybe can we talk about that now? For those that are, in the midst of completing the quantitative results, you could just bear in mind that your other accordingly help describe what they're seeing. So let's start with chem. Chemistry. And the idea is that we had 5 different ingredients. We had 20 degrees, we had 30 degrees, we had 10. So we had 10 degrees, second degree 20, 30 degrees, 37, 39, and then 60. So what? Almost, yeah. So 10, 20, 10 degrees, 20 degrees, 30, and then 39, 57, and then 60. What are your results looking like? I'm going to start from increasing the data. And everyone, let's pay attention. Limit discussions for now because you want to take note of these observations. Have a table in your full class for today, describing or making note of what others are saying. So what I mean is, you're going to have a table describing the different or the scale of different categories. So let's say the markers aren't too well. So the idea is that you have temperature as one table going at each increment. So 10, 20, 30, 40, and then 60. I want you for each of the bacterial to be able to use the scale that I'm referring to from 0 to 3. If you did like earlier 0 to 5, that's okay. But I want us to go over that right now. So that way it'll make comparisons thereafter. For all 4 bacteria, 5. Everyone take notes because these are values you want to record. You need to get a new member in each group to be able to record these values, so that way you can make note of them. So all group members should have already recorded scale for all different turbidity across. And so we'll talk about that now as a group, so that way you guys can record it for each can I take your balance?
It makes sense.
So, if you want to come to the front, you can. It's very simple. It's not like a major presentation, but folks, I need you to all pay attention. But you need someone to say it out loud so everyone else can hear you. They need to record the values. So I prefer just a person comes and says, B subtilis, 3 out of 3. E. coli, 2 out of 3. Our temperature, and that's it. You don't have to worry. You don't have to say intro, discussion. I mean, if you want to, you can. But essentially, this is your final, like, subtle presentation. And you just need one person to talk about it. So don't worry. Starring Ashwin, group 1. Your temperature is
10 degrees.
10 degrees. Everyone start taking note. Okay. Begin. Your scale 0 to 3, right?
Yeah.
Okay.
E. coli, Phyllis, B and B subtilis and control were all 0. You got
Say it one more time. Control, B subtilis.
control, B subtilis, the S
The B stereothermophilus. All of them were 0.
E. coli were 0, but we got M's Phyllis as 3.
What was the last one? Microbiology?
Yeah, that was the one that we got 3. So that showed turbidity.
Okay.
And the other 4 did not.
Okay.
And then for the B, the same thing happened. E. coli, S, B thermophilus, B subtilis, and control at 0. And then for Phyllis, we got 3.
Okay. So one other point I want to highlight, you will have to do this for the other tables as well. So just bear that in mind. You can also confirm thereafter, but it's better that you come, have one person in each group come forth, describe what they did, the temperature or the condition for each of them, temperature, salinity, and pH. You won't have a table for them all. And I recommend to make it easier, just say it for one rep if they're all the same. Remember though, when you come up to the front, remind us your group, their temperature, like each condition that you have for each of the exercises, temp, pH, salinity, and then thereafter describe the bacterial absorbance, the qualitative observations that you have, mentioning the scale as well, if you did 0 to 5, so people know accordingly. With that, good job. Oh, sorry, pH and salinity. We need to grab that as well. Can you mention that as well? Salinity.
The effective
Start with pH. Which pH did you use? pH 2, right? pH 2.
And then for all of them we got 0. For S epidermis, we'll say, and all 3 got 0.
So okay, now your salinity.
Slow down.
Okay, how about we pause just for a moment briefly. In your tables, have one that's temp, one pH, one salinity for all the different conditions. And what you're doing is very simple because you're just putting numbers, right? So you have your axis here, like your one column here, describing the different bacteria, right? B. subtilis, E. coli, Staphylococcus, epidermidis. And then here, your condition of 10 degrees, 20 degrees, 30 degrees, 40 or 39 degrees, and 60 degrees. Thereafter, by the way, it's actually 37 degrees, so 37 and then 60. The idea is that you're just putting like numbers, 0, 1, 2, 3. You could do 0 out of 3, 0 out of 4, 0 out of 2, whatever scale that you use. We can, but the issue is how are we going to get that data?
Oh, we have to start saying
It's on the board.
Okay. You know what? I'm just thinking qualitatively and efficiently. What's the time? 11:08. I don't think we have the time. So let's see. I prefer people listening, but I understand. Yeah. Okay. You should have the charts already made because it'll make sense to just add zeros. Okay, so how about this? Okay, if you need time to make a chart, then that's okay. How about this? We have a few options. Make a chart. Send me the data by tonight because the reason why I'm cognizant of this is because I noticed some of you haven't done so, and it just makes it difficult for those that are waiting. I don't want us to waste time. So let's do this. Make your tables for the time being, for the next 3 minutes, and then I hope that you'll be able to say the values easily. Everything okay? I don't want you guys to feel down. You don't have to feel down. I want you guys to have the opportunity to actually hear other people's voices, to actually, record the data accordingly and, feel like you're able to be the next TA of this course. So, that being said, if there's questions, let me just see something. I'm going to make note of a small adjustment. Give me a moment. Is that the question as well? Is that your question as well? Give me a moment. Okay, sharing the data? Yes. for spec 20s. That's okay. That's okay. That's okay. That's okay. Which bacteria is this? Okay. This should be okay. Folks, are we all done the spec 20s, by the way? All of us? One group is not done? Okay. In the meantime, hopefully that's 3 minutes. If you guys have a table, that's amazing. Don't worry if, sorry, just the observed data. I mean, the spec 20s would be good as well. You should say the spec 20s. And this is what we're going to do, folks. Each person can spend maybe a minute or two just talking about it in their group. And then by that time, we should be done at 11:24, right? So Ashwin, if you can, sorry, you can come up just saying what you've done. I think I have another student either named Ashwin or Ashwin as well. So, but nonetheless, what I want us to do, let's record the data. That makes sense? Okay. That's okay. That's okay. Folks, come up, don't worry. And pretty much just write them down what you hear from people. And it's okay if they're not if you have questions, you can ask even via email, hopefully. Can I have group 1 come? Let me just say actually what they said for temp. 10 degrees, and correct me if I'm mistaken, all of them were 0 except for Chirophilus, S. Staphylococcus chirophilus. That was a 3 out of 3 for temp. All of them for pH at 2 was 0, so 0 out of 3. And then they now have to mention salinity for absorbance and qualitative readings. You can say it from there. How about that? Yeah. Can you guys all hear? Zara, can you hear? Melina? He's about to speak. Yeah. All 0. Okay, okay. Okay, say it out loud. Start off with salinity. What was yours? Oh, your bacteria. Yes, your bacteria. Staphylococcus epidermidis for 2%, 5%, 8%, and 11%. What are yours? 1 out of 3. So 1 out of 3 turbidity readings. Absorbance. 0.628. So now we're at spec 20 results. Try to write it down. Don't worry, you guys. It's not too difficult, trust me. And I didn't. So it's probably not. Okay, don't worry about this. Take the FLR aside for now. Let's listen to what others have to say. Let's be mindful of their results. Now, once again, can you repeat, Ashwin, your spec 20 results? He's mentioned them already for qualitative. Now quantitative, yes. 2%? Can you say it again for 2%? Staphylococcus? Okay, 0.628, 2%. 0.476 at 5%. 0.638 for 8%. Okay, okay. 0.482. 0.482 for 11%. Great job. Next group. Group 2 will now speak about their temp readings, all qualitative for temp, pH, salinity. And it will take a bit of time with the markers that may not. Yeah. It's okay. let's take our time to talk about it just briefly.
So we were doing 20 degrees.
20 degrees, folks. 2 out of 3. Out of 3, yeah. Okay. The sterile thermophilus. Okay. So remember, at a scale of 0 to 3, can you just repeat it again for your first one?
That was 0. And for
Subtilis?
B subtilis was 1. Our control was 0.
Okay. So does anyone need that to be repeated?
Which one do you need?
Okay. Okay. Perfect.
pH 4 it was all 0 as well.
Okay. And then salinity.
So for our salinity, we did S epidermis as well, like with 1. We've got 1 for turbidity across.
Okay.
And then for our absorbance reading for S epidermis, we got for 2%, 0.708.
0.078.
708.
0.078.
No, 0.708.
Sorry, 0.708. Yes. Yes, yes, yes. Sorry, my mind was. Yes.
For 5%, we got 0.465.
0.4. Sorry, say it one more time. 465, 5%.
8% was 0.484.
484, 8%.
Yep. And 10%, we got 0.522.
0.522, 11%. I'm just. Great job. You mentioned the salinity as well, right? You mentioned it for salinity of the qualitative, right? Just
Yeah,
because and likewise for pH. 1 across. Okay, 0 across for pH across. Salinity 1 across, 1 out of 3. Great job. Next group. Can you guys share your results? Can you say it a bit loud, folks? Let's pay attention.
For the full line, the prior one and the subtotal are all 0. And then for the long name, that one is 2. Okay, this scale of 5.
Okay.
But a 5 or 0.
Okay, so let's give context here. Yeah, so pretty much say a 1 out of 3. It's technically 0.8, but we'll say 1 out of 3. But this is actually why I prefer larger ranges. So you guys are able to see differences, like a 1 versus a 2 out of 5, etc. Continue though.
Cafe one goes for 0. And then the serpentine one is 3.
Does anyone need any results to be repeated? Slower. Okay, temperature one more time. I'll try to echo them, so we'll take time.
Okay. B subtilis and then prior one were all 0.
Okay.
And then the long name that was 1.
So that's the only one that had 1 out of 3. Okay, but technically you had 4 different bacteria, right? So I heard.
Yeah, so all of them were 0.
Oh, they did.
No, we have 4 bacteria.
Only B. Sorry, continue. Okay, that makes sense. Okay, so all of them were 0 except for B sterile thermophilus or sterile thermophilus. That's a 1 out of 3. Yeah. Okay, so E. coli was a 1 out of 3. B sterile thermophilus was a 1 out of 3.
So B sterile.
No. B sterile thermophilus. The thermophilus E. coli are 1 out of 3. You know what? I feel like this may sound a bit confusing for you guys, unless you guys are able to record the values. Are you able to? Yeah? This is okay? Okay. Otherwise, and this is my kind request, please, please. If you're gonna send them to me, please send them to me today because I am often waiting, and the sad reality is that other people are waiting for their post lab and they find out like the day before, and that's not fair, right? For their reporting of results. So if we do this to make it easier for you guys, for the tonsils as well, or like for your understanding, we could pause for now.
Well, made our charts go.
Everyone's good? Jacob, are you good?
One question share results with you that has everybody.
That would be amazing. Okay, let's continue then. pH one more time. Okay.
F 0, L passing was 0, Cerviciae was, and our
Everyone understood pH?
so yeah.
Yes.
Or okay, so we did 2% 500 so all of yeah, for turbidity, all of them zero, all the percentages, they're all zero. And then for absorbance, 2% was 0.015. 0.015. 5% was 0.007.
0.007? Okay. So
8% was
could you remind me of your bacteria?
30.
8% you said or 5%? Was it 5% at 0.007 or 8?
5.
Okay.
And then 8% was 0.012.
0.012?
And then 11. 11% was 0.004.
0.004. Okay. Group 4, which is now a group of 6 people, can they share their results? They're 39. I'm just gonna complete something else in the meantime. So keep saying them out loud. If you want to come to the front, you can. Okay. Okay. Complete everything. Like say everything that you are for now. Someone else. Are you on the last one for salinity? That's good. Okay. So record the remaining. Like say what you have to present, and in real time, maybe Garvin's gonna tell you the final meeting. So you can begin. Just say it whenever you're ready. Can you say a bit louder?
And prior to 0 as well? Yes.
Okay, next one, pH. pH 8, okay. Okay. L-casein?
Yeah.
Okay. 1 out of 3, you said?
Yeah.
Okay.
That's 0.
Okay, that's good. Continue. 0 out of 3 for the last one.
You did yeast. And for that one, for all the concentrations, we had 1 out of
Okay. I think he's done right now though, right? How many though? I thought you said one. Okay. So don't worry, that's okay. We still have a bit of time, so we'll move to group 5. Don't worry, that's okay. Um, but I would recommend, as he's doing it by himself, let's see if we can help him out. That way, like at least you'll be able to double the effort and reduce the time by half. So hopefully, try to help him out, help Garvin out. One of you, Muhammad, Saad Rahman, grab the other one, help him out accordingly. Now, Melina, group 6, Scott, Karis, Alta, Jacob. Are you in group 5 or 6? Okay. No problem. And then Zara. Now you guys can present your temp, 60 degrees, pH, I believe it's 8, 11, 10, and then there are details for salinity. Whenever you're ready.
So the temperature was 60 degrees, and both controls were 0 out of 3. I think I said that right. Both of them were 0 out of 3. The sterotheranophiles. E. coli was also 0 out of 3. Everything was 0 out of 3.
Everything? Okay. Feel free to summarize it again.
Yeah. Is everybody good with that? Okay. pH of 10. Everything was 0 out of 3 except for S. epidermidis, which was 1 out of 3.
So Staphylococcus epidermidis is a 1 out of 3. Everything else was a 0. For pH, right? Okay.
Salinity. We did E. coli, and for 2%, 5%, and 8%, it was 3 out of 3, and for 11%, it was 2 out of 3. And
yes, last one was 12. Can you repeat the bacteria?
E. coli.
E. coli. E. coli, 2 out of 3. Everything else, 3 out of 3.
And then
Everything else was a 3 out of 3 for pH at 11. Sorry, for salinity, everything was a 3 out of 3 except that for E. coli at 11% it was a 2 out of 3. So yeah.
And then for spec reading, 2% 0.800. At 5%, we had 0.845.
0.845.
At 8%, we had 0.840.
0.840.
And then finally at 11%, we had 0.835.
0.835. Okay, so that's the last one at 11%. So we'll briefly wait. Don't worry, folks. Well, as we confirm a few other things, let me just briefly talk about classification of bacteria. So you have different environmental conditions we've talked about: temperature, pH, and salinity across a variety of different conditions, right? We went from 2 to 11% in salinity. We went from 10 degrees to 60 degrees in temperature. And I believe for the pH, we went from 2 to 10%, pH 2 to pH 10. So, and that in turn actually refers to concentrations of hydromium or hydrogen ions. So that's also another thing to keep in mind. These are now, think of it as media, liquids in which now you're inserting bacteria into. And so by them being able to grow, that depicts their level of not just propensity in that specific environment, but also their ability to essentially reach their optimal growth rate. And so as you can see from the different curves on this slide, that there's a wide variety of trends and a typical curve that is bell-shaped, right? Like a normal distribution. But there are cases like for temperature where it's more skewed, like a right hand skewed. So you want to bear that in mind. It's like towards the left, it's skewed downwards. But the idea is that this is often replicated across different bacterial groups. And that's shown here in temperature quite neatly. Sacrophiles are like cryophiles in a sense. Cryophiles are referred to their specific bacteria, like I believe we had Micrococcus cryophilus. And that turn refers to a more cold attracted bacteria, which is indeed an example of a saccharophile. You have hyperthermophiles, like the ones that you see in hot springs, where you probably would get Taq polymerase, so that's something to keep in mind. And that's where you have B styrothermophilus, which is thermophilus essentially, so it likes hot temperatures. And so that's what you want to keep in mind. There are different ranges where you have psychotrophs and thermophiles that are more towards the mesophile range, so they're more closer than the further extremes on either end. And then you have pHs. And by the way, with that in mind, you should be able to actually determine quite easily what you would expect for turbidity readings, even if you were just to give this example, let's say at a lab exam. So just think about like these different, terms, the different root words, the suffixes, prefixes, etc. Right? And then for pH, you have acidophiles, more on the low pH range. You have basophiles or alkaphiles, which are on the high pH range, and you have mesophiles that are generally at the middle pH of 7, 6 to 8 range, which is more physiologically. Well, actually, the beautiful part of our bodies is that we have different ranges of pHs, right? We have like the cerebrospinal fluid that is like a pH of like 5 to 6, more towards the mesophile range, and then you have your gut flora, which is where, at least in the stomach lining, for H. Helicobacter pylori, which if you think of ulcers and other gastrointestinal conditions, they are more towards the acidophile range, so like lower pH ranges. And that has important relevance, not just for your lab exam, but if you're interested in med school, research, whatever it may be, these different bacterial have novel conditions for you to optimally grow them. And that may be useful for your own interest in discovering different proteins, drugs, and different inhibitors that may be useful down the road. Now, after that, you have osmotic pressure. An example is what we've seen from osmolarity. The idea is that if you increase concentrations of NaCl, it's a table salt, it's a solute. And the interesting point is that chlorine is a halogen, right? So this connects us to chemistry because you have halotolerant, you have halophiles and non-halophiles, which is similar to what we've talked about before for non-osmotolerant tolerant versus osmotolerant bacteria. The idea is that if they're osmotolerant, they're able to grow at a wide range of concentrations of solutes, or essentially, let's say, E. coli hypothetically, would grow from 0 to 8%, right, of NaCl. And that also reflects the type of solute, right, because they're different halogens, they're different solutes indeed. So like magnesium chloride, you have table salt, sodium chloride, you have other ones as well that you want to keep in mind. And then like halophiles are within usually the middle range, similar to like a mesophile. And then you have the extreme halophiles that are more towards high concentrations of table salt in this regard, or you have the non-halo tolerant, which is more towards the non-induced or the limited amount of solute present in that vicinity. So non-halophiles are similar to non-osmotolerant bacteria. And that's where you have E. coli versus Staphylococcus epidermidis. And beautifully noted is that you have gram-positive versus gram-negative bacteria, right? And we know the difference, peptoglycan layers. And that thickness also reflects the effects that they would have therein when exposed to high concentrations of NaCl and other solutes as well. Before you leave, there's one final exercise, which I'm going to condense. So that way you probably can leave early, I hope. But I'm going to describe things on the slide because it may be difficult to see on the board. We're going to the last page, page 12, and you don't have, to my understanding, you have some discussion questions that we talked about before, but I want you to think about this now, connecting back to the past with regeneration time assignment. So if you recall, your log graphs, semi-log graphs went upwards and then maybe it led to a plateau, right, over time. In this regard, you're actually doing it the opposite way. way, because as you imagine, as you increase temperatures for specific bacteria, if they reach their extremes, noting that each bacteria has an optimal range of thermal growth, if you are exceeding that range, then you're eventually leading to reduced bacterial growth. Or in this regard, essentially, you would find over time a decrease in the number of viable bacteria, which connects us with your plates. So from your plates, we have the microplate data already on Quercus, but for your plates, what I need you to do, and this is what you could send me via email, is your final CFUs for your specific condition. So we were 4 groups, remember? 50 degrees at 0 minutes, 50 degrees at 5 minutes. Sorry, I switched it up. 50 degrees at that side, 65 degrees here, and then we had 0 at 0 minutes and at 5 minutes, right? Recall thermal death time, we used these 2 beakers and I was like doing secret operations with the microplate. So the idea is that when you're looking now at your results, what you need to do is the following. You are gonna have, remember, you had 6 plates. From the plates that are within range of 30 to 300 colonies, you're going to count them up. Let's say your dilution factor for a plate that was in that range was 10 to the negative 5, and you had 121 colonies. So with that, you would have 1.21 times 10 to the power of 7 CFUs per mil, right? So essentially, take the dilution factor, remind yourself you're connecting back to the past with the plaque assay you calculated, right? Likely the CFUs per mil, or the PFUs in that regard. And so in this regard, you're going to calculate your CFUs for your plates within range of 30 to 300. If you have 2 plates or more, then you're going to take the average and record that and send it to me. What you need to do is when you're done that, and I'll hopefully send out that once you're done that, you'll have 2 curves because you only have 2 time points, right? 0 to 5 minutes. So the range is not as large, but you should have 2 curves, one that's depicted at 50 degrees and another at 65 degrees. And so the goal is for each curve at a specific temperature, you're going to connect the dot through 5 minutes, and you should be able to see the trajectory go down. If it doesn't, because I'll hopefully see the results beforehand, I'll give you data that you hopefully will be able to use. But nonetheless, I would expect and hope that we would see a decrease over colonial growth. If you find that not to be the case, and let's say this comes up as a lab exam question, you need to think of your methods. Think about any prospects of contamination, errors in volumes, right, or resuspension. These are 3 common concerns and limitations that I want to think about. And going back to this graph, once you're done plotting, just like what you did for generation time, you're going to be now taking 2 different stats. Once you've made a curve and you have a line, you're going to take the TDT. TDT, or thermal death time, refers to the amount of time it takes for your bacteria to essentially reach 0 in log, right? Technically speaking, won't really reach 0, but like when you plot it, you'll extrapolate that line, so probably go up to here, and then you want to let that line go all the way down to where it reaches the x-axis, and from where it began to where it ends is your thermal death time. So let's say it's 90 minutes where it reaches, and it may vary, maybe less likely based on what you see and based on what you've recorded on the semi-long paper, which you can make manually. Like on your graphs, on your in your lab notebook, like as a sketch, or you can actually print out a sample like what we've done from generation time assignment. That's one stat. And let me know if I need to slow down. But the D decimal reduction value, that's where essentially, and it's more simple, you're going from let's say 10,000 CFUs to 1,000. So you're decreasing by 10-fold. So it's like find your initial volume to be at the top, which makes sense for us because we have values at 0 minutes. You can start off from there and see where it would lead to going 10-fold under. So for example, what I mean by this is someone's Bunsen burner is on too high. By the way, please lower it. I'm getting like reactive Sorry. Oh, good. Let's go back to that idea. I said 1.21 times 10 to the power 7 TFUs. If that was our T0, let's say 50 degrees, then what you need to do is go down to 1.2 times 10 to the power 6, right? You're going down 10 times from 10 to the power 7, 10 to the power 6. And so when you're down there, you want to compare either at 0 or from another location, which is shown here, where it's like 10,000 midway to the curve, or somewhere in between. What I want you to do is essentially go from the top, go down to where it reaches tenfold, and essentially deduce the difference from 0 to that end, or essentially that final x value. So interpolate, right? 1.2 times 10 to the power 6. Go down and see what that time range is, and that's your d value. I'll ask a question. Because we're dealing with two curves, you may want to think about this. At 50 degrees versus 65 degrees of E. coli growing, at either time point, do you expect there to be a higher thermal death time or lower for 50 or 65? Sorry, which one would be a higher thermal death time? Actually, lower, to make it easier. Which would be easier to... Let me sit down. Okay. Because I've spoken... Okay. Thermal death time, right? Takes longer. You want to observe the amount of time it takes for your bacteria to become or to pass away. And the idea is between the two temperatures, which one would have a higher TDT value? 50 or 65 degrees? 50. Why? That's correct. And the reason I ask why is not to like intimidate anyone, but these are questions are often parts of what you have to answer in your lab exam. You can't always simply say yes or no. You have to explain. So, and that's a great response. There is one caveat that you may want to be mindful of. As we talked about before, there are ranges. So you beautifully pointed out, right, for E. coli, you want to bear in mind the other ranges of bacteria. Hypothetically, if we had a cryophilus, it would be similar. But if you had thermophilus, at 65 degrees, you would probably see a lower TDT, sorry, a higher TDT value, meaning that it would take longer for it to kill at high temps or longer to pass away. So that's like, okay, to make it simpler, TDT reflects the time it takes for your bacteria to die or to pass away. And then D reduction values are also similar, but they're for a finite amount of time. And they're reducing your population by 90%, which is why it goes down from 100,000, for example, to 10,000, right? Because now that reduction is by 90,000, right? So it goes down tenfold, which is 90%. So now if we couple that knowledge, 90%, and by the way, brief reminder, folks, pay attention. Please also make sure you're not using your phones without letting me know. Make sure your lab coats are buttoned up. Okay. You're taking a photo of value, sorry? Okay, that's amazing. I appreciate it. Sorry, a bit of a tangent, but let me just highlight this now. Once you once you get your curves, inshallah, you're going to get those stats. And D value is very similar to the TDT, that if you're at high, for our case, E. coli, 50 degrees, you should see either a higher or lower D value. What do you guys think? Higher or lower? For 50 degrees, should you see a higher or lower D value? Lower? At 50 degrees? The TDT and D values should be similar in trends. Higher for both. So higher for both. Just to wrap your heads around this, it's the opposite of generation time. So the longer it takes to die, the less sensitive it is at that temperature, right? So bear that in mind. That reflects or the longer it takes to pass. So bear that in mind. TDT reflects the total time. or is it essentially, sorry, Bunsen burner is off, right? No? Okay, it's okay. TDT, D values, record those values. If you have questions, let me know. That is the end. Before you leave, can I just confirm everyone has recorded those values for their colonies? That's the end of the slides per se, but you need to take those recordings. Make sure that you send me those values as soon as you can before you leave. When you're done, setup includes the following. And I actually have a few other things, so we do have time. If you have asked me before, then you can go. But I just need you to all pay attention to the final announcements that I have. Clean up your benches, shut off the spec 20s with the left bottom knob. You just invert all the way to the very bottom where it says 0. Yeah. So just keep going down. It should eventually shut off. Make sure as well you put your trays. Great question. So you place your blue trays into a single rack here. So there's technically space for two, but try to place all your tubes once you're done recording the values. Once you've taken even photos of your results, make sure that you place the tubes down here. And then once again, make sure that for you have everything. That's great. Honestly, any used oil, the paper towels, any extra waste, just make sure that you place them either into the Wescadine or for the autoclave for paper. There are two other things, or maybe I'll condense it to one. So FLRs, let me see, is there anything else? Okay, post-lab, you guys should be aware of, right? Follow the same protocol as before. Generation, actually unknown assignment. Please grab your unknown assignment. Unknown assignment. The Bunsen burner is still on. Sorry. Unknown assignment. If you can grab it, that'd be amazing. And briefly, just to highlight FLRs, I already told you guys what you need to do, and so hopefully you should be able to prep up for your next one. Do on the April 2nd. So bear that in mind, as well as the timing. I believe it's at 10 p.m. And so we already have the values, right? Also on purpose. So you can use the figure that's already posted there to describe what you've done essentially during that lab. Remember to take note of what I've described. As I'm a bit tight on time, I haven't been able to give everyone detailed feedback. However, if you have seen my purpose announcement and you find that there are things that maybe you're missing out and want further clarification, please let me know as early as you can. Just bear in mind, you may have to send repeat emails because I am a bit busy or occupied with other classes as well. But with that, I wish you guys all the best. We're going to be at our last practical next week. But remember, folks, there's still a bit that you can do, right? So if you're like thinking about, oh, what's my performance right now? Think about what you've done thus far in terms of your lab notebooks. Make sure that once again, flow charts, your results, figure captions, discussions with the questions that we've discussed here in the slides and from the manual, that you've done them in detail. And also making sure that as you come back for our last week, that you have everything that you are ready for to not only complete the lab, but to be able to be well prepped and well ready for the lab exam. Because you have many things that you likely have already prepared for. And now it's just really the time for you to make use of the final day, our final week, to discuss any outstanding questions for the final exam. And also Dr. Brunt will have likely prep up for you guys for the lab exam. So just stay in tune with purpose announcements. And if there's any follow-up questions or announcements, you'll likely see them soon. Take care, everyone. If there's any questions, let me know. Thanks again. So happy to have you guys finish well in advance. Keep it up. Once again, test tubes, all there. Try to place them in a single rack if possible. If there's not space, they can also be left right outside. There is like you see that blue wrap? Yeah, there's space. Perfect. So try to just make use of the space that you have. Let me see, anything else? Yeah, that's it. I don't think there's anything else. I just want to make sure. If you guys want to send me your values, you can also let me know them now. I can record them directly for your mean viable counts as well. Yeah. With that, have an amazing week, everyone. No problem. Perfect. I can actually note it. This is 9.8 times 10 to the power of 8. I think it's a bit large, actually. So
But
that,
the only one that shows was this, unless the dilution factor I'm doing this wrong.
this should be 10 to the power of 7.
So, you make sure you turn the gas off.
Yes. Thank you, Dr. Martin.
Is it-
So yes. Let me confirm. Actually, I'm gonna write it down. Which yes. Yes. So now the thing is about the reason is 0.1 mils. It should be 0.1. But is this not entirely? So, by the way, just to clarify as well, or sorry, like to answer the question, this should be 10 to the power -7. Like it would be 9.8 times 10 to the power 7, because this should be your final dilution for your plate, right? So if you do 98 divided by 10 to the power 6, then it will be 10 to the, so I'll record it as, yes.
I thought it was the other way.
Oh, okay, I understand. But this isn't for YouTube, this is for your plate, right?
Yes.
Perfect. So one last question. What is your condition? 50 degrees at 5 minutes?
Yeah.
Perfect. Okay. If any other groups have their values, I could take note of them now, in fact, for CFUs. Can I actually get your values? I could get it from each group. Carlos, can you grab groups as well, viable counts? Okay, can you ask if that's okay? Okay. Oh, sorry, curious, was there another question? Give me one. Yes.
The way that each phone is a different.
I would do it at the final. The final should be okay. Thank you. Yes, I do. If there isn't a change in absorbance, go with the first time point. But you're only doing one time, so you don't have to worry about all of them. And the thing as well is that they are just at 50 and 65 degrees for the microplate, so you won't have to worry about changes in time. Actually, wait, sorry. It will be, sorry. I think you understand. It's across time, smaller intervals. But that should be okay. Any other questions?
So after 2 hours time for all of the.
Actually, do it across time, and you only need to do it for one. Oh, sorry. 120, yes. Like I'll look at it once again just to confirm, because each row had a specific time point. And that's why I was just thinking about the incubation time, which is also a separate confounding variable. So essentially go with it in a specific table at a specific time point, and that should suffice. Just before I do, I need to answer to you. Is that the final?
Yeah.
Are you sure it's 7? And that's 2.73. Okay. That's the mean for 1 or 2 plates. Okay. Great job. You were playing it in one. Technically, it should fill up.
Yeah.
How are you guys doing?
Great.
I'm gonna send it to. I can even write it down. Amazing. But for your Bible.
My Bible, the only one we had was -5.
Okay.
Was 156 colonies.
And
The
your
rest of them was the first two, the one below
now you said this is at T0 65 degrees. Take care.
no. Does that make sense? 65? Me check.
No problem. Let's have a look. Or like remind me email please, because I need to it may take more time than I can. Like I wouldn't be able to probably give you full justice. But let me know. Okay, do you recall? Did you look at the breakdown?
Yeah. Like the email.
If you, normally I send. I didn't mention it, but have a look
Like
on the grade book. Do you have a comment section?
Yeah, but there were.
Click on the desktop because I normally send comments. And I actually try to break it down for each category. So intro, results, discussion, because otherwise it would be more qualitative, and I want to make it consistent, or at least I want to be like here marking. A separate point. Actually, give me time to. Is that okay?
Yeah.
We can talk about it. Is that feel free to remind me or email me, please, if that's okay.
Oh.
So that way I could describe more details if you need more comments. Maybe because there's a lot of students I marked, so I don't know. But let me know. Is that who had a question I missed? Mohammed.saeed@utoronto.ca. Carlos. Okay. Okay. Let me just also say this, because I know probably some of you are wondering, okay, what happened to the FLR? I normally send a breakdown, so I didn't know they were published. I mean, it's good that you guys find out, but I do want to give you guys some. I already sent a quick message detailing specific details on what was or what I expected. Bear in mind that there are specific things that are expected for each component that are highlighted either in the outline or what I've discussed in lab. I also want to highlight that I generally try to deliver guidance justly and fairly. So even though this is, okay, I didn't say this, but I normally scale up grades. So that's outside of like the TA contract, but essentially I try to see, okay, let's say because I'm following a rubric, right? A guideline, right? Of how like I'm supposed to mark. Let's say hypothetically the grade average is 68. I normally up it to about 75 around that sense, like maybe 7% up. And that's really to be not only fair, but to be just and mindful of you guys, right? Because I understand that there are some like nuances, things that maybe are difficult to understand. Maybe Dr. Bunt has highlighted, or maybe I highlighted, but maybe you weren't able to hear, or maybe you didn't like fully understand. So I try to be like aware of that, and or at least try to compensate for any ambiguities by upping the mark essentially. That was done also for the unknown assignment. So I generally will try to do this, but I want you guys to also understand that there is time and like there's sufficient time for you guys to do these assignments. And I normally try to do them out of my, I try to give you guys more time than other labs. As you're probably aware, you guys had an extra week, if not two, during reading week to look at it a bit. So I hope that you guys are able to accordingly reflect on what I've talked about. If you want me to give you specific comments, let me know. I'm more than happy to. It's just because I have like a lot of reports, it's hard to essentially go through everyone and provide them the same amount of space when I only have a finite amount of window, actually like a finite amount of time to like I want to just to be very honest with you guys, I want to make sure that I do my part, but please also do the same, right? Because you have that time. You had almost like a month to actually do this FLR. And this one is now the one upcoming is like maybe for 2 weeks time. So see what you could do, gain the insight that you need, and feel free to email me, come by next week, ask me questions, come by to the Student Center, you may see me there, and I'll be more than happy to talk further with you guys. I want you guys to all shine brighter than the sun, right? So like brighter than these illuminating ceramic lamps, but just do your best and leave the rest with the best. That's all I can say, folks. Once again, I hope you enjoy the rest of the practical. Let me know if you have any questions. Final point about lab performance. I've done something in the past and I'm going to make it easier for you guys, or maybe not easier, but understand where I'm going with this. Just like how we had gradients, right? Like your performance. I go from, so hypothetically, I could go from like a TA could go from 0 to 100% in lab performance, right? Right? I will go from 60 to 100%, meaning that essentially, I honestly guarantee that you pass, but guarantee that I at least do my best to distinguish those that really want to get a 4.0, right? And those that are just doing what they can. Because I also understand maybe you're having 5 other lab courses. So I want you to understand that. Now I'm going up in increments from 60 to 70 to 80 to 90 to 100. I may adjust this, but the point is that if you are hypothetically at an 84, right? So the threshold is an 85 to be up to a 90. If you show me next week, and even this week, with answering questions, sending your FLR, sorry, you could do your FLR as well, but asking me questions, sending me your pre-lab, post-lab in advance, and showcasing that you're demonstrating like, full equity, like full like excellence in this lab, you'll likely get a 90. So like boosting it up from there. If like, actually that's all I can really say, because I want to make sure that I'm also fair. If like hypothetically you've done amazing in your lab notebooks, clear, organized, condensed with the details of discussion questions, those likely will reflect your performance as well. But I just want you to also bear in mind that as I'm just, I also try to be fair, and in reverse, as I'm fair, I also try to be supportive for you guys. So just do your best in this course, and I hope you guys will do your best and shine brighter than stars in the night sky. So with that, take care, folks. If there's any follow-up questions, let me know. Take care and have an amazing week. If there's any final questions, concerns, let me know. You may prefer to do it via email as well. That's fine, yours, right? No. I'm trying to think who has headphones. We can leave them inside. They may email as well. Anyone recall those headphones? Do they belong to anyone here? No? Yeah? Okay, so we'll leave it back to where it was, just in case that student comes back into the same cabinet. Hopefully they'll grab it. Thank you so much. Both of you. No problem. Take care. Take care, you guys. Absorbance. Oh, we didn't talk about it. Okay, send it to me.
We did.
That was the last one. Or what did you get?
For like all of them, or just
If you have them in memory, I'll write them down.
okay, I have them in the book. For the temperature value, only two came up. Highest, that was in 0.7 or 4 for A, 0.76 for B, Subtilis, second highest, 0.12, 2.12, and everything else.
And this is for just the temp.
This is for the temperature, we want the zinc. Okay. Everything goes zero except interest. This is for salt concentration. Surprisingly, all of them are roughly about 0.0. That's a zero thing. Yeah, I did have this.
So, okay. Oh, sorry. I thought this connected with the huge. Okay. So
Yeah, I don't know.
that's okay. I mean, what our goal is to actually see across the bacterial streams, the species, to see how they fare at a specific concentration as well. But I wanted to organize the so that way it's more efficient. That way they don't have to worry about going to different tubes and essentially it makes things easier for people. We had it from before, but hopefully inshallah keep those values and focus. If not, just be able to describe everything from your values and see whether or not it actually makes sense. Do you think, for example, then this interprets your actually what I would recommend is seeing what you can classify there for the different bacteria. So be it across a specific range, but any specific community, pH, and you could be able to see generally speaking. And you have as well turbidity as a